Okay. Food, structure and bonding in food molecules. Proteins and amino acids, fats and lipids, carbohydrates. So this is the last three areas. And then energy of food is to finish. Proteins, right? Key knowledge area. Formation of dipeptides and polypeptides. Di means? Poly means? Many. As condensation, what's that? Produces water. Polymers of two amino acids or alpha amino acids. 2 stands for the second carbon in which the amino functional group comes off, because off the number 1 carbon is the carboxyl group. Yeah? Primary, including peptide links. Secondary, tertiary and quaternary structures and bonding. We'll talk about that. Distinction between essential and non-essential amino acids as dietary components. So what I want you to do as part of today is that get out your best friends, and we're going to be doing some annotating of said best friend, right? If you've got coloured pencils, I encourage you to get them out for said task. Carbohydrates we'll talk about in a future lesson. Formation of disaccharides from monosaccharides, complex carbohydrates, condensation polymerase of monosaccharides. Mono means 1, thank you. Glycosidic links or ether links as they're also called. Storage of excess glucose in the body as glycogen, comparison of glucose, fructose, sucrose and the artificial sweetener aspartame with reference to their structures and energy content that we will explore later. Fats and oils, triglycerides. We've seen triglycerides before. Where have we seen those? Biodiesel, right? So we're coming almost full circle. So we start with a fat and oil to make methyl fatty esters. In digestion, we make fatty acids rather than fatty esters. So we're using water as the hydrolyzing agent, not methanol. Distinction between fats and oils. Anyone want to have a guess at what the difference between a fat and an oil is? Well, state for one. What's that? Fat is solid, oils are liquid. But what are they? Well, they're both triglycerides. Yeah? So what do you think? Length of the chain. Yeah. Explanation of different melting points with reference to structure of their fatty acid tails and strength of intermolecular forces. So the longer the chain, the stronger the force of attraction. More dispersion forces, right? Because they're largely non-polar. Chemical structures of saturated, what's that? No double bonds, yep. Unsaturated, double bonds. Mono unsaturated would mean what? One what? One unsaturated. One double bond. So polyunsaturated would be? More than one. Yep. Yeah, distinction between essential and non-essential. Unlikely to be triple bonds, but it's more likely to be doubles. Distinction between essential and non-essential. What does the word essential mean? Important, right? Non-essential means? Right, so slightly different definitions in relation to fatty acids. Essential means we can only get them from our diet. Non-essential means we can synthesize them in our own bodies. Structural difference between omega. What does omega mean? Last, right? So omega-3 might mean what? Third from the last. Yeah? So omega-3 fatty acids and omega-6 fatty acids means there is a double bond, 3 carbons from the end. Omega-6 means there's a double bond, 6 carbons from the end. Yeah? Nah. See, when you boil down the chemistry of it, right? Omega-3, omega-6 fatty acids, not that interesting. Vitamins, inability for humans to synthesize most vitamins except vitamin D. Where do we get vitamin D from? Sun, yep. Vitamin D is a cholesterol-based vitamin. So, interaction between the sun, manipulating cholesterol. It's also a number of your hormones are based off cholesterol as well. So, cholesterol is actually quite important. My mum did her honours thesis relating to high and low density lipoprotein, and the reason why we don't, I think her study looked at the reason as to why we can't use rabbits for the study of cholesterol. So this is 1975? 1975. She did her research on dietetics. And strangely enough, unethically now, her human sample was her own blood. Yes, she used as a human sample in her research. Comparison of structural features of vitamin C, illustrative water-soluble vitamin versus vitamin D, fat-soluble. What might determine the difference between fat versus water-soluble? Polarity, thank you. Excellent. All right. Let's take a step back. Condensation reactions. As we said, condensation is the formation of bonds. So we've got to think condensation is the formation of bonds. What's the opposite of condensation? Hydrolysis is the breaking of bonds. Condensation has water as a byproduct. How many waters are produced? One, yeah, so or n minus 1, where n is the number of components. Yeah. Hydrolysis of triglycerides, we've seen that before. Difference being is that rather than using methanol as our hydrolyzing agent, we're using water when talking about digestion. Please do not drink methanol. Why? Heavily poison it. Okay, it reacts with in the body to form formaldehyde. Formaldehyde attacks the optic nerve and literally makes you go blind. Due to the polarity of the functional group, attacks something about the nerve. Yep, something. Did I get to 5 yet? the 5 whys? Whatever it is. Oh no! Eventually, something about evolution will get we'll get to as to why those interactions will not work. Okay. Proteins. Woo-hoo! Proteins fundamental to cell structure and operation. Cell structure of plants is slightly different, but the cell wall is actually carbohydrate based. Proteins, otherwise, for most cells, important to cell structure and operation. Built from smaller monomer. Mer, right? Mer means unit, right? The suffix of mer means unit. Mono means one. So monomer is one unit. Polymer means many units put together. Amino acids. So every amino acid has an amino group, NH2, and a carboxy group, COOH. Carboxyl, carboxy, same. Yeah. Amino acids have the general formula H2NCHR. Now, in this context, R is not necessarily a alkyl chain as per proton NMR, but R is the rest of, or R is the R group, which can change. Yeah, so it could be alkyl, it could be functional, etc. List of common amino acids on page 14 or 15 of your best friends. Types of protein. Don't really need to know types, but I guess it's helpful to understand the importance of proteins in our body. Structural, protection, support, movement, skin, bone, cartilage, ligaments, tendons, muscles, hair, teeth, feathers, beaks, cocoons, insect exoskeletons are all protein-based. Bone is also slightly ionic-based as well. Yeah? Lots of the others, proteins. Enzymes, biological catalysts, often protein-based. Yeah? We also can have what's called a coenzyme. Biologists in the room, what's a coenzyme?
It's a compound that's not an enzyme or a protein that helps an enzyme function. So it's like a non-organic.
Cool. It's co as in cooperate. It cooperates with proteins to assist with the function of the protein. Yeah? And in terms of enzymes, a coenzyme is something that interlocks, and we'll get to that anyway later. Transport, movement of compounds between and within cells, like hemoglobin, is a quaternary protein made up of myoglobin subproteins. Protective, defense, antibodies, proteins, toxins, attacks, snake and spider venoms, proteins. Most proteins are also an energy source. Notice it says only in extreme circumstances. So your body will initially break down all the glycogen, then start to break down the fat, and then once the fat starts to break down, your muscle fibers and your protein starts to break down as your energy source. Yeah? So that's why anorexia nervosa, you get that gaunt look because you get muscle wastage. Yeah, or same thing with terminal cancer patients. They start to literally look wasted because the muscle tissue is being broken down. Right? Cancer cells are energy intense cell tissue. They suck in energy like crazy. Yeah. Look at that. First ever photograph of a protein. Well, they're two separate photos. Yeah, shows hemoglobin proteins. New technique. Find out more here if you're interested. Amino acids. There's a structure. There's the amino group, carboxyl group, CH, and the R. And we'll talk about the different R's. Yeah, often referred to as alpha amino acids or 2 amino acids. 2 carbons. Amino coming off the second carbon because this has priority of 1. Make sense? So we're not looking at 3 amino acids, right? We can get other amino acids that are longer, but we're only looking at 2 amino acids in this course. Different amino acids as different groups of atoms in the R group. 2, 1, 2. Amino coming off the second carbon. Yep. So like with our nomenclature and naming structure, carboxyl has the first priority. Amino's coming off the second carbon, 2 amino. Some simple examples. Glycine, all of our amino acids we explore, and you'll notice in your table, have a 3-letter abbreviation. So glycine, gly. This is glycine. It's the simplest of all the amino acids. Your amino acids are listed in alphabetical order, not in terms of simplicity. So in glycine, the R group is an H. Alanine, which should be close to your top, has a methyl group as its R group. Does that make sense? Glycine is the simplest because it's got the simplest, right? Simplest R group, being H. This is the next simplest, being a CH3. Any questions about that? So just notice and be mindful of where some of those abbreviations are similar, right? Like glutamine versus glutamic acid, and different. Yeah, so just be mindful of that. Asparagine versus aspartic acid are different. Probably. I think there's a form of urea or uric acid or whatever that causes that strong ammonium smell. Yeah. Don't go telling too many people that. Okay. Here we go. Here they all are. Yeah. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, right? Different. GLU versus GLN. Glycine, histidine. Yeah. Histidine. Isoleucine, leucine. Iso meaning like isomer. So we've got the same molecular formula of the R group, just in a different configuration. Leucine, lysine, methionine, phenylalanine, right? One of the main components of NutraSweet. So have you heard of phenylketonuria? No, that's something completely different. phenylalanine. You've got to be really careful. Some people, I think it's 1 in 15,000 people, you'll notice on the back of diet soft drinks will say contains phenylketonuretix. So that means it contains phenylalanine, which is part of NutraSweet, part of an artificial sweetener. 1 in 15,000 people cannot process that amino acid. All right, so phenylketonuria, you can get tested for this early on. Right from birth, you can be tested on this. It's part of the heel prick test, I believe, which tests for this. It's a rare but really important condition because it means that you have to have a low protein diet for the rest of your life. So no bulking for people that suffer from that condition. It's pretty rare. Proline. Interesting thing about proline, the R group. Yeah, as you notice, the R group actually loops back around and connects to the amine. So just be mindful of that. Serine, threonine, tryptophan. Who's heard of tryptophan before? Where have you heard about it? Okay, where's a more common place where you might find tryptophan? Apart from a data booklet. Anyone heard of turkey? The food. Apparently. No, it's not really what makes you sleepy. It's been attributed, it's in Turkey, as in the meat, not the country. Tryptophan is attributed to making you sleepy, but it's more likely that the sleepiness is caused from overeating.
We learned about a specific gene in prokaryotes that produces tryptophan and self-regulates levels.
I really want to hear about that. Tyrosine and valine. Okay, so 9 essential amino acids. So this is where our annotation of our data booklet begins. Obviously, you can't take in a annotated data book into your SAC or examination, but if we're annotating it and looking at it the whole time with this unit, then we're going to start to get used to where and what we're looking for in these different amino acids. So our 9 essential amino acids are histidine, isoleucine, leucine, lysine, methionine, phenylalanine. So from leucine through phenylalanine. Why are they essential? Remember, essential means your body cannot synthesize it. So it must be in your diet. Yeah, so the only source is from your diet. Right, and then skip a couple, threonine, tryptophan, skip tyrosine and valine. They are our essential amino acids. Nine essential. There's 9 there. Don't think I've got that wrong. Steroids aren't protein. Non-steroid steroids could be cholesterol based or similar. Steroids are generally not protein based. Cool. Do I need that up anymore? I can go back if need be. Okay, here's an activity. I'll pause the recording, see if you can guess them. You can use a pencil if you like, and then we'll go through the solutions. Using different colours, identify the acidic, the basic, the sulfur, polar, and non-polar R groups. So the R group is the bit that comes up here, right? Difference is the R group in proline is here, it goes down as opposed to up. All right, so I'll leave it there. I'm going to pause the recording. Can I pause recording?
Really?